In this, our third and final lesson from Chapter 11, we're going to finish talking about how traits get inherited from one generation to another. But in this lesson, we're going to look at things that Mendel did not discover. Basically because these are things that didn't show up in the pea plants and honeybees that Mendel was experimenting on. But these are things we've discovered since his time that don't violate Mendel's laws at all, but just are some things that kind of are a little different than what he experienced in his experiments. And one thing we found out is that there are some cases where actually blending really does exist. Exactly what Mendel was kind of setting out to prove was wrong, sometimes does happen. Sometimes, if an individual is heterozygous, it doesn't just show one form of the trait, it actually shows a blending of the two. And when that happens, we call it incomplete dominance. We can look here in our picture, we have a red flower and a white flower, and even though we're using capital and lowercase letters, really neither trait is dominant over the other. So when we cross the red flower and the white flower, we get pink flowers in the F1 generation. Now the symbols we use to show incomplete dominance are not standardized. So sometimes you'll see people using just the regular capital and lowercase letters. Sometimes someone may use R and R for red and W for white and R and W for pink. There's no standardized form here. Incomplete dominance is definitely rarer than just a normal dominant relationship. But what really gets strange is when you cross two of those F1 plants. If you cross two of these pink flowers with each other, what you end up seeing is a 1 to 2 to 1 ratio in their offspring, because crossing two pink plants gives you a 1 in 4 chance of a red flower or a 1 in 4 chance of a white flower. So suddenly now we have a single trait that can have 3 different phenotypes, and it gets a little bit confusing if you only know about the basic dominant and recessive ideas that Mendel had discussed. Another thing we've discovered is sometimes heterozygous individuals show both alleles simultaneously. We call this codominance. And in codominance, instead of crossing a red and white flower and getting a pink flower, we can cross a red flower and a white flower and get a red and white flower, where both traits are showing up simultaneously instead of blending together. Now, incomplete dominance and codominance work very similarly to each other. They both are going to work out the same in Punnett squares. We'd see again a 1 to 2 to 1 ratio if we would cross this flower in the picture with another one similar to it. They're so similar that sometimes some people get them confused. Sometimes you'll even see people grouping the two together and calling them the same thing. But there is a difference here. In incomplete dominance, neither trait completely dominates the other, whereas in codominance, both traits are dominant and showing up in the heterozygous form. But again, for both cases, we don't have a standard way of symbolizing it with letters, so you see all sorts of different things. Sometimes you'll see a letter C with a little superscript letter next to it to show codominance, and sometimes people even use the same letter C when they're doing incomplete dominance. Well, a third thing we've discovered that Mendel didn't know about is the fact that some traits don't just have 2 alleles for that trait. Sometimes a trait may have more than 2 alleles. And the first place where this was discovered was actually in human blood typing, just the basic letter that makes up your blood type, whether you're A, B, O, or AB blood type. This is caused by having more than 2 alleles for this particular trait. So another way of saying that is the gene for your blood type has more than 2 different possibilities that can exist there in that locus on the chromosome. Now the 3 alleles actually are what those 3 letters are. We're going to talk more about blood typing when we get to our immune systems later on this year, because that's actually when blood typing ends up being important. But in terms of the inheritance, there are 3 different alleles, and A and B are co-dominant alleles, meaning if you're heterozygous with both A and B, they'll both show up. That's how you get blood type AB. O is recessive, so the only way you can have type O blood is if you have 2 O alleles. Now I want to emphasize here, more than 2 alleles exist. There are the A, B, and O alleles, but still for each person, because you have 2 parents, you still only would get 2 alleles for each trait. It's not like you'd get 3 alleles handed down to you. You only have 2, but there are 3 possible alleles from each parent that you could get. Now, we're going to look at this a couple different ways because they love to ask about this on the AP exam. This chart maybe looks a little confusing at first, but on the left-hand side here, it's saying, not for every person, but this is a possibility of what you could get from your father. You could get an A allele, a B, or an O. Again, because he only has two, there are at most two possibilities that your father could give to you, but for all humanity, there are three possible alleles that a father could give. They're in the same way three possible alleles a mother could give. But then in the white boxes, they're showing what your blood type would be. If each parent gives you an A, it makes sense you'd have A. If each parent gives an O, as we said before, you have type O. But if your dad gives an A, your mom gives a B, you're going to have type AB. Because O is recessive, if your dad gives an A and your mom gives an O, you're only going to have type A because O is recessive to A. So we've got all these combinations, but to me the easiest way to make sense out of this is to go right to a Punnett square and look at a problem. So let's do a problem where we have one parent with type O blood and another parent with type AB blood. Now, if the first parent has type O blood and O is recessive, we know they have two recessive alleles. The typical symbols we use when we're doing human blood type is we use the letter I as the base letter. Don't ask me why. I've tried to investigate why. I've found a few different answers. Probably the best answer is that at one point they thought this was an incomplete dominance description here, so the letter I for incomplete. But if you have type O blood, you'd have two recessive alleles, so we use lowercase i's. And if you have blood type AB, you have two codominant alleles. So we're going to have a dominant, capital I with a superscript A, and a dominant I with superscript B to show those two different dominant alleles. Now, even though we see four letters here, those superscripts go with the I's, so really it's only two alleles we're describing. Think of it as two letters. Well, if we make up our Punnett square, the first parent can only give a recessive allele. The second parent can either give the A allele or the B allele. So when we fill in the boxes, let's do the first column first. Those offspring would have type A blood because A dominates over O. The other two possibilities are offspring that would have type B blood. So this is kind of weird because if you have one parent with type O and another parent with type AB, none of their children can have a blood type that matches what the parent has in this case. So you see some really unusual relationships when you just throw in that one extra allele over having the normal two for a trait. Okay, some other stuff that Mendel didn't discover, and again, probably because his details on his experiments stopped after just the basics of inheritance. He did not understand the fact that sometimes you need more than one gene pair to really determine what your trait is. We see this especially in fairly complex organisms. We call these traits epistatic traits, or we say the condition of having more than one gene pair to determine a trait being epistasis. And what happens is one gene pair somehow seems to affect the expression of another gene pair. And we found this in a few different situations, especially in traits that seem to have a lot of variation to them. One specific place where we found it is in mammal hair color. And of course, since humans are mammals, this includes us. And specifically one place where we found it in humans, according to our current understanding, we believe there's one gene pair that determines whether or not you have red hair. You have another gene pair on a completely different chromosome that determines what shade your hair is. So for the first gene pair, if it determines you don't have red hair, the second gene pair then seems to determine whether you are blonde or brunette. Now, if that first gene pair determined that you should have red hair, the second gene pair may determine what shade of red hair, whether it's a lighter or darker shade of red. Now, the problem is we don't have a full explanation there for all the different varieties of blonde and browns and blacks that exist. So we're assuming this may be even a case of more than two gene pairs that exist. There may be three or even four gene pairs. There seem to be multiple proteins involved in that hair coloration. So it gives you a lot of different phenotypes that are possible. So in hair color for mammals, we don't just see two things. It's not like in the pea plant where the peas were either yellow or green. Another thing Mendel didn't discover is something called Playa Trophy, which in a way is kind of like the opposite of epistasis. In Playa Trophy, One single gene pair, one single locus on a chromosome, can affect two different things about your phenotype. The first case we discovered this in humans was with what's called sickle cell and the gene for that trait. Now, sickle cell is usually described as a disease that affects people of African ancestry. And there's a recessive trait. If you have two copies of the allele for this trait, it gives your hemoglobin a weird shape, and it makes your red blood cells have a different shape. They're shaped kind of like a crescent moon shape, or what was often described first as a sickle, which is a type of tool that's not used very often anymore. And so if you have the sickle cell gene from each of your parents, then you would end up with this disease, which is often a deadly disease. However, we also found that if you have just a single copy of the gene, it gives you immunity to malaria. And so since you only need a single copy, that's a dominant thing. So the shape of your hemoglobin, immunity to malaria, two different traits, two different parts of your phenotype, one recessive, one dominant, but both coming from a single gene. Now, the last thing that we believe Mendel would have maybe eventually discovered if he kept experimenting, but did not come across, is the fact that we seem to be more than just what our genes make us up to be. The environment plays a major role also. And it seems like every year we're hearing more and more about how the environment is affecting how our genes are expressed. And so it doesn't have to just be your external environment, which is what we usually think of when we hear that word. It can be your internal environment as well. Outside temperature or the temperature of your body, either one, can possibly affect how your genes are expressed. Foods you eat, just several different factors around may change the way a gene is expressed or whether or not a gene is expressed. In humans, we see this with our eye color, we see it with our hair color, height. You know, at the most basic level, you can even think of like your skin color, whether or not you're tanning, as being a bit of influence from the environment. And I think in the future, we're going to see even more relating environment to how our genes are expressed. That's the end of chapter 11. Bring your questions to class, and we are going to move on to more advanced levels of genetics after this. So have a great day.